Hello, my name is Cindy Scavin, and I'm a product specialist in CBER's Division of Cellular and Gene Therapies. My seminar will focus on topics that manufacturers may encounter as they advance to later stages of product development. The goal of my presentation is to help guide you towards successful development of quality cell and gene therapy products. Successful development and approval of all biologics requires that the product be safe, pure, potent, effective, and stable. In addition, you must demonstrate control of the manufacturing process and the continued ability to manufacture a quality product after licensure. Manufacturing control is demonstrated by product consistency and current good manufacturing practices, or CGMPs. Product consistency is demonstrated through testing and characterization of product quality attributes throughout the product's life cycle. It's important to recognize, however, that quality cannot be tested into products, but should be built in by design and should include thorough evaluation of the manufacturing process at all phases of development. A quality product attribute is a product characteristic selected for its ability to ensure the suitability of the product for its intended use. It may include, for example, physical, molecular, or biological properties of the product. Collectively, the quality attributes define the safety, purity, potency, identity, and stability of the product. Understanding your product's quality attributes is critical to successful product development. Biological products are complex, often heterogeneous mixtures with complicated mechanisms of action. This complexity underscores the need for product consistency, which is necessary to generate solid clinical data. The more you know about your product, the more straightforward it will be to determine the relationship between certain product quality attributes and safety and efficacy. These relationships will be critical as your product advances towards licensure. The data obtained for product lots used for safety and efficacy studies will be used to support specifications for lot release and product storage. The relationship between product quality attributes and safety and efficacy will also be important when developing methods to show comparability between product lots after manufacturing changes. While it's not expected that all product characterization activities be met prior to starting phase 1 clinical studies, we encourage you to begin evaluating product quality attributes during early product development. Expectations for product characterization change during a product's life cycle. Preclinical and CMC safety testing and basic product and process characterization data should be provided prior to initiation of phase 1 studies. This information is covered briefly on my next slide. As the steps and arrows in the diagram indicate, product development activities may be implemented in incremental steps, but should progress along with the phase of clinical development. For your BLA, your product should be fully characterized and comply with the biologics regulations outlined in Title 21, Part 610 of the Code of Federal Regulations. In addition, process control and CGMPs, as outlined in Parts 210 and 211, should also progress along with clinical development, as shown by the arrows on this slide. This slide summarizes the CMC safety and basic product characterization information that may be expected prior to a phase 1 study. Briefly, your product should be tested for basic safety parameters, including sterility, endotoxin, and contaminating viruses. While a validated potency assay is not necessary, you will need to determine the strength of your product for dosing purposes. You will also need to determine potential impurities related to raw materials and/or process contaminants. And while full compliance with identity standards may not be required to initiate phase 1 studies, it's sensible to have an assay to uniquely identify your product, especially where multiple products are being manufactured in the same facility. Please refer to the web seminar titled CMC for Cell Therapy and CMC for Gene Therapy for more in-depth presentation on these issues. The rest of my presentation will focus on product characterization in the context of CMC development lifecycle. During early product development, tests are performed to collect data about product composition, biological activity, and other critical product attributes to help you better understand your product. We recommend that you evaluate a number of assays to gain as much information about product quality attributes as is feasible. Product characterization activities may also help you determine product quality attributes related to stability, for comparability assessments following manufacturing changes, and to evaluate in-process controls during manufacturing. A subset of these assays may form the basis for the testing used to meet compliance standards for product release as outlined in Part 610. As product development advances, you will need to demonstrate control of the manufacturing process through adherence to CGMPs. Compliance with CGMPs is a critical aspect of controlling product quality through process control and validation. In addition to outlining facility and equipment controls, CGMPs also define requirements for setting specifications and acceptance limits, as well as the requirements for assay control and validation. Tests used for lot release specifications need to be qualified or validated depending on the stage of product development. CGMPs are defined as a set of current scientifically sound methods, practices, or principles that are implemented and documented during product development and production to ensure consistent manufacture of safe, pure, and potent products. For Phase 1 studies, cell and gene therapy products are exempt from CGMP's regulations outlined in Part 211. However, to ensure reproducible, consistent performance of product lots, general CGMP principles are applicable for all phases of clinical study, including Phase 1 products. Some CGMPs should also be in place prior to production of clinical lots to ensure product safety. These include sterility assurance, quality oversight, and facility controls. Adequate documentation, including well-defined written procedures and controls and data from manufacturing, including testing, should also be in place. For more information on CGMPs for Phase 1 studies, please refer to the Guidance for Industry, CGMPs for Phase 1 Investigational Drugs, published in July 2008. These principles are also outlined in the OCGT web seminar on CMC for gene therapy products. Because CGMPs apply to both the manufacturing process and the facilities, product manufacturing and characterization and CGMP processes cannot be separated, but must develop together. However, similar to product characterization, certain CGMP activities may be implemented in incremental steps. I'll discuss a few of these examples on my next slide. It's expected that control of the manufacturing process will increase along with clinical trial advancement and should overlap with your understanding of critical product quality attributes. You should be aware that for Phase 2 and 3 clinical studies, Part 211 applies to cell and gene therapy products. It's recommended that you refer to the FDA guidance on the preparation of IND products published in 1991 and evaluate process controls during early product development. Some examples of CGMP activities that may develop as you progress with clinical development include process improvements and validation. This might include activities such as optimizing transduction efficiency or purification of a cell population. You may also be working on methods development and validation, such as determining the robustness of an assay or improving the sensitivity or specificity of a PCR assay. You may also continue to develop and monitor process controls and set specifications based on knowledge of the manufacturing process and product attributes related to safety and efficacy. We also recommend that you arrange a Type C meeting with CBER's Office of Compliance and Biologics Quality, especially with the Division of Manufacturing and Product Quality, when making decisions related to facility design, manufacturing controls, and process validation. I'd like to take a minute here to discuss lot release specifications. Specifications are the test and acceptance criteria that confirm the quality of products and other materials used in the production of a product. They can include both analytical and biological procedures. Acceptance criteria are the numerical limits, ranges, or other criteria for the test described in specifications. Product specifications may develop in incremental steps over the course of your product's life cycle. For instance, during early Phase 1 and 2 clinical studies, you should have specifications in place for safety parameters with defined limits. However, the acceptance limits for other product characterization assays, such as biological activity and purity, may have wider ranges since manufacturing data may be limited. As you advance in clinical development, you should be determining which product quality attributes are related to safety and efficacy and narrowing the acceptance limits for assays used to measure these attributes. By Phase 3, you should have well-characterized tests with meaningful acceptance limits in place so that you have confidence that the product lots used in these studies will be expected to be of similar quality. For your BLA, the assays used for final product lot release specifications must demonstrate accuracy, sensitivity, specificity, and reproducibility through validation and should include solid statistical analyses as outlined in the CGMPs. ICH guidance document Q6B describes recommendations for setting specifications for biotechnology and biological products. While this guidance document was not written for cell and gene therapy products in mind, the general principles outlined can be applied. In accordance with CGMPs, validated assays should be used when testing licensed products for release, stability, and when comparing product lots following manufacturing changes. Methods validation is a process of demonstrating that analytical procedures are suitable for their intended use. This involves making sure that an analytical procedure does what it's intended to do, yields data to answer a specific question, and provides confidence in the results. As discussed on the next slide, assay validation requirements are defined in the CGMP regulations. An FDA guidance document, Analytical Procedures and Methods Validation, published in 2000, and the ICH guideline Q2R1, Validation of Analytical Procedures, published in 2005, have been developed to help manufacturers meet validation requirements. We also recommend you consult with your CBER review team. Method development and validation are aspects of CGMPs that can be implemented incrementally during clinical development. However, assays used to evaluate specifications for characterizing licensed biologics must be fully CGMP compliant, as described in Parts 210 and 211. Briefly, assay results must be available prior to lot release. Results should be quantitative and meet predefined acceptance criteria. Lot release assays should also include a comparison to an appropriate reference material or control when available and must be validated for licensure. We recommend that you consult with a statistician prior to method validation. In addition to complying with CGMPs, biologics must also meet product standards described in Part 610, which include tests to measure potency, purity, and identity. Potency is defined in Part 600.3 as follows. The word potency is interpreted to mean the specific ability or capacity of the product to affect a given result. Tests for potency are defined in Part 610.10, which states that tests for potency shall consist of either in vitro or in vivo tests, or both, which have been specifically designed for each product so as to indicate its potency. Due to the complex nature of cell and gene therapy products, development of suitable tests to measure potency can be one of the more challenging aspects of product development. To help manufacturers meet potency requirements for INDs and BLAs, the FDA published the Draft Guidance for Industry Potency Tests for Cellular and Gene Therapy Products in October 2008. As outlined in the guidance, all licensed biologics, including cell and gene therapy products, must meet CGMP requirements for assay performance as previously outlined. In addition, your potency assay should also indicate the biological activity specific or relevant to the product. The assay should also measure activity of all ingredients considered necessary for activity. We understand that with some complex biologics, there may be some ambiguity as to what components of the product are critical for activity. However, if you plan to claim an ingredient is necessary for activity, you will need to develop a test to evaluate that activity. The assay used to measure potency should also be stability indicating, meaning it should be able to detect product degradation or loss of other quality attributes. While it's not expected that all assay requirements will be met prior to starting a Phase 1 clinical study, we encourage you to start potency assay development and product characterization activities during early product development. It is expected that potency assay development and implementation should progress along with clinical development and should be refined as part of an incremental approach to product development. During early product development phases, we expect you to characterize your product quality attributes and investigate the mechanism of action to justify a critical biological activity or activities as a target for potency assay development. We recommend that you evaluate a variety of assays to measure product attributes related to the mechanism of action. One or more of the tests you evaluate may have sufficient robustness to be validated for lot release specifications. As clinical development continues, we expect your potency assay to be refined and qualified as suitable for use. For pivotal and late phase studies, you should have an assay in place that's based on product lots shown to be safe and effective during previous phases of clinical development. A validated assay should be described in the BLA with acceptance criteria that reflects potency limits for product lots shown to be safe and effective in pivotal studies. Potency assays are based on product-specific attributes. Therefore, there are no set expectations for which assay method should be used to demonstrate potency for cell and gene therapy products. However, since cell and gene therapy products are biologics, they should have demonstrated biological activity. Outlined in this slide are a number of approaches that could be used to measure potency. You may directly measure biological activity using an in vitro or in vivo bioassay based on product-specific attributes. This is the traditional method for measuring potency for biologics. For some products, a bioassay may not be feasible. In such cases, you may also develop an indirect measure of biological activity using an analytical method or methods that measure immunochemical, molecular, physical, and/or biochemical properties of the product, in which can be correlated to the relevant and specific biological activity. This is called a surrogate measure of biological activity. It's also possible that one assay may not be sufficient to demonstrate potency. In this case, a combination of complementary assays or assay matrix may be developed that measure the relevant product quality attributes necessary to demonstrate potency. Please refer to the guidance on potency tests for cell and gene therapy products for a more detailed discussion on these approaches. Due to numerous challenges developing potency tests for some cell and gene therapy products, it may not always be possible or feasible to use a bioassay for lot release. In such circumstances, an analytical assay or assays may be used as a surrogate indicator of biological activity, provided the methods are substantiated by correlation to a relevant biological activity. Correlations should include sufficient statistically sound data and may include a comparison to preclinical proof of concept data, to in vivo animal or clinical data, or to in vitro cellular or biochemical data. Because a significant amount of data may be required to support correlations, we recommend that you initiate product characterization studies during early product development. Product purity is another product standard outlined in Part 610 that must be described in your BLA. This standard states that products should be free of extraneous material except when it is unavoidable due to the manufacturing process described in the approved BLA. You should carefully examine the impurity profile of your product and provide a description of process materials that are considered unavoidable due to the manufacturing process. You should evaluate risk posed by process impurities as part of process development and validation, as outlined in my next couple slides. During early CMC development, the final product should be tested for contaminants or impurities that may be introduced during production. In addition, tests to monitor process controls during production should also be developed as early as possible. As CMC development progresses, the manufacturing process should be validated to remove or not introduce process-related contaminants. Unsafe contaminants should be removed, and for other impurities, limits should be set based on data from lots shown to be safe in preclinical or clinical studies. It's important to note, however, that if process impurities are not sufficiently characterized in product lots used in early phase preclinical or clinical safety studies, it will be difficult to set safe limits, and impurities may confound your ability to evaluate product safety and efficacy. When considering tests to evaluate a product's impurity profile and to evaluate process validation, you should consider several factors. One is the capacity of the process to remove manufacturing components, such as media components and helper viruses, or product isoforms such as empty particles or unintended cell populations. You should also consider the type of process control procedures you have in place for the purification system. How well are reagents qualified? What do you know about your process? What in-process tests do you have in place? Is equipment single-use? Will columns be single-use or regenerated? These and other questions will help you evaluate the risk that contaminants might be introduced during the purification process and to evaluate the safety risk associated with possible contaminants, such as cesium chloride or other chromatography material. Identity testing is described in Part 610.14. It states that the final container of each lot should be tested for identity after all labeling operations have been completed. The identity test should be able to identify the product as designated on the final container, package label, and circulars. The test should also be able to distinguish the product from any other product being processed in the same facility. We recommend that manufacturers evaluate multiple assays during product development to characterize unique product attributes for identity testing. In addition, demonstrating that products are free of facility contaminants will be necessary to meet cGMP compliance. Let's consider an identity assay for a gene therapy viral vector that's produced in a facility that manufactures multiple gene therapy vectors. A manufacturer wants to use a simple PCR assay directed to the transgene for identity. However, consider the following potential problems. 1. The components used to produce the vector may also have the transgene, for example, the plasmids, chimeric helper viruses, or indicated proviruses. 2. Other products may have the same transgene but be on different backbones. For example, facilities makes an adenovirus and a herpes virus. And 3. Products may have the same genome, express the same transgene, but have multiple serotypes. This example illustrates how solid understanding of the unique properties of your product will be necessary to allow you to distinguish your product from possible contaminants or other products manufactured in the same facility. Stability testing for products under IND is described under Part 312.23. Demonstrating product stability is required at all phases of clinical development because you must be able to show that the product is within acceptable quality limits for the duration of your planned clinical study. We also allow an incremental approach for stability testing and setting limits and ranges. Data to support stability of your product for Phase 1 clinical study can be based on data from preclinical lots that have been stored in the same manner as the clinical material. For example, they have the same formulation, dilution, temperature, and container. By Phase 2, you should have refined and tightened limits and ranges based on data collected for lots used in Phase 1. For Phase 3, you should be collecting data to support expiration dates and storage conditions. For the BLA, you should have collected data using validated assays with preset acceptance limits. Stability testing for licensed products is described in the CGMP regulations under Part 211.165. You should have a written testing program in place which is designed to assess the stability characteristics of your drug product. This information is used to determine storage conditions and expiration dates. You should also refer to the ICH guideline Q5C published in July 1996 for additional details related to stability testing. Briefly, as part of process control and validation, your stability testing protocol should also include an evaluation of stability during planned storage conditions using the planned formulation. You may also want to evaluate more than one formulation during early development if stability may be an issue. You should also evaluate stability during manufacturing holding steps and under stress conditions. You may want to consider the potential commercial and operational implications of long-term storage and shelf life at certain temperatures, such as -70 degrees Celsius. You also need to evaluate shipping and transport conditions, as these will need to be validated for licensure. And finally, you will want to evaluate the stability of the product after loading into the delivery device. This should be done at all phases of development to ensure adequate dosing of an active product. Shown here is a partial list of potential process changes you might encounter during product development, such as process changes, changes in materials, scale-up, a new manufacturing site, formulation changes, introduction of quality elements such as DNA testing, and changes in analytical procedures. Because change is part of product development, it's important to remember that even small changes in manufacturing can result in unpredictable changes in safety, efficacy, antigenicity, biodistribution, and/or stability. It's hard to predict how even minor components or contaminants might be important to safety and efficacy, which is why product characterization is so important. In addition, because product consistency is so important for interpretation of clinical data, we recommend that you evaluate your process throughout development and avoid making major manufacturing changes while in Phase 3, as this could result in the need for significant data to demonstrate comparability between product lots used in pivotal studies. Unfortunately, there is not enough time in this presentation to thoroughly cover comparability issues. However, there is guidance available on how to determine comparability following manufacturing changes. Please consult ICH Q5E, Comparability of Biotechnological and Biological Products Subject to Changes in Their Manufacturing Process, published in June 2005. And please consult with your CBER review team prior to making major manufacturing changes that could potentially affect product safety or efficacy. I'll emphasize again how important product characterization is to understanding the relationship between product quality attributes and safety and efficacy. I'd like to summarize by emphasizing that all aspects of product development, including process development, manufacturing control, research activities, analytical tests, stability testing, biological tests, and lot release testing, are all interrelated, with product characterization being central to your understanding of what the product can do. Is the product safe? Is it effective? And can you consistently manufacture a quality product? These are all necessary elements for successful development of safe and effective cell and gene therapy products. I've included a general contact information on this slide. For questions regarding this webinar, please contact me at denise.gavin@fda.hhs.gov. And for general regulatory questions, you can contact the regulatory management staff at CBER OCGT RMS, all one word, at fda.hhs.gov or by phone at 301-827-5102. All CBER guidance documents can be found on the website listed on this slide or by following links from www.fda.gov. Thank you for your attention.